This conference will now be recorded.
So when we are talking about taxonomy of a plant, you start with K. K is kingdom, then
Phylum.
followed by phylum, very good. Then
Class.
KP, cough, COF, class, very good. Then
Order.
Order.
order,
Family.
family,
Genus. Species.
genus, species. Okay. So although when we are studying the individual drugs according to your syllabus, we don't go for kingdom or the remaining KP, cough, whatever I told you. We don't go for kingdom, phylum, class, order. But you should know the family, you should know the genus, and you should know the species. Clear? So in Cassia angustifolia, Cassia is the genus and angustifolia is the species. Is it clear? Okay, now coming back to the pharmacognostic scheme. So the first one is synonym. Please write down in your books, put today's date, write down unit 2 and just write the heading as secondary metabolites. Below that you write pharmacognostic scheme of a medicinal plant. In that first one is synonym. Next is, after synonym is
Biological source.
biological source. Biological source has 4 parts now. One is it tells about the condition, it tells about the plant part, it tells about the genus and species of the plant, and it also tells about the family, and most important one, it also tells how much amount of main active constituent should be present. Like for example, I'll tell you the biological source of coriander. We'll tell coriander consists of dried ripe fruits of Coriandrum sativum belonging to family Umbelliferae, and main you'll tell it should contain not less than some 0.3% of D-linalool. Clear? This is the details what you give in biological source. So how many details are covered now? Yes, how many? What all you have in biological source? You have the plant part. I told you it is dried ripe fruits. So dried is nothing but the condition. Next is ripe fruit. So dried ripe is the condition. Fruit is the plant part. Okay. Coriandrum sativum becomes the botanical name. There's a difference between botanical name and biological source. Botanical name just means genus and species, whereas biological source contains the complete details. Okay, so it consists of dried ripe fruits of Coriandrum sativum belonging to family Umbelliferae. It should contain not less than 0.1% of D-linalool. So what all details a biological source has now?
Condition of the plants.
Condition. Then
Family name.
don't jumble it, in order you tell me. Condition of the plant.
Part of the plant.
Different part of the plant.
Part of the plant, good.
And family name.
Botanical name, genus and species.
Contents of the active constituents present.
Family name and the limit of main active constituent which must be present. Now what happens if, see why that is very important is, if the limit of the main active constituent is less, then of course, what does it mean if the limit of the main active constituent only is less? If your drug, whatever you have taken for analysis, does not obey the limit as given in standard books, what does that mean?
Every plant is differed in limit of the active constituent.
It clearly means that the drug is substandard. Okay, it is not official. Clear? The drug is substandard. So that's why limit of one constituent only will be given, which is the main constituent. Now, as we know, plants have more than 100 constituents. But why 1 is important here? Based on that, we are going to make the final preparation, final pharmacological preparation. Clear?
Yes, ma'am.
Okay.
Yes, ma'am.
So first is synonym. Second is biological source with family. Third one is, after that, what do we study?
Morphology, ma'am.
Good. Morphology. So in morphology, how do you study? At the left side, you're going to, the left side, you're going to draw the diagram. And what details you will be writing? Quickly.
Structure, color and all. Shape,
There,
size.
there is a pattern for that. I have taught you in the first initial classes. What order you should follow? What you said is right. Color is fine. Shape is fine. What else? And in what order?
Microscopic structure.
No, no. Now we are discussing about morphology. Let us finish morphology and then go for microscopy. In
Yes, ma'am.
morphology, what all do you study? What is the other name for morphology? Color, odor.
Organoleptic. Color, odor, size, shape, and special features.
Very good, Yashanjali. So we are going to follow this order. COT, C-O-T. Color, odor, taste, shape, size, extra features. Now, when you all are giving presentation, you should give it in this order only. First is synonym, then is biological source, then comes morphology. You can take any image from Google which is pertaining to that particular plant. You can insert an image there, but you will be writing in the order as color, odor, taste, shape, size, extra features. What do you mean by extra features?
For texture, ma'am.
Texture, surface, not area, surface.
Smell of
Yes.
the, how it smells.
Uh, any other additional features, like for example, if you're taking leaf, about the midrib, about the venation, about the apex, about the base, isn't it? If you're taking a fruit, you can tell what other features you can observe, how many lines you can observe, how is the fruit surface, how is the epicarp, pericarp. If you're taking a bark, you can tell outer surface, whether it is rough, whether it has fissures, whether it has wrinkles. Inner surface, you can tell whether what is the color of the inner surface, whether it has striations, that is lines, etc. So all these are in extra features. So what are the other name for morphological evaluation?
Organoleptic.
Organoleptic, or it is also called as sensory evaluation. What is it called as?
Sensory evaluation.
Sensory evaluation. Even you call it as macroscopic evaluation. Macro means what? What do you mean by macro?
Large or visible, ma'am.
Something which is bigger, opposite of microscopy. So next will be microscopy, which you're going to observe under microscope. So organoleptic is nothing but macroscopic evaluation. Why organoleptic or sensory evaluation? Because you're using your sense organs to analyze it. You're using your eye, nose, skin, isn't it? You're using all your 5 sense organs to evaluate it. Clear? Okay, what is it? So first one is synonym. Next is biological source with family. Then comes morphology in the order of color, odor, taste, shape, size, extra features. Then comes, what's the next thing which you'll study?
Microscopic evaluation.
Microscopic.
Very good. Microscopical evaluation. Now, I have told you in the initial classes, microscopy can be done by 4 different ways. Okay. So, according to you, microscopic analysis means what? Yes. Microscopic evaluation means what you will do. Come on, answer whatever you know. Even if it's wrong, fine, no problem.
Observing under a microscope.
You're going to observe under microscope. What will you observe?
The features.
You can't keep
Cells.
the whole plant part as such. You
Yes,
can't keep the whole plant part as such. So what do you
Yes.
do?
Yes.
Very good. You're going to take the transfer section. Okay. I could hear even somebody telling powder. Very good. So you either take the powder and you observe under microscope, or you take a transfer section of the plant part and you observe under microscope. Very good. There are two more things what you can do in microscopy. One is leaf constants. There's something called as leaf constant, which is very, very important, and it gives you certain parameter where you can identify a particular crude drug. Okay, so that also can be analyzed by quantitative microscopy. So quantitative microscopy, have you done last year in your practical? Determination of starch, stomatal number and all?
On
yes,
Yes,
ma'am.
ma'am.
stomatal number.
Okay, so that is nothing but quantitative microscopy. And the last one is we can also find out percentage purity of the drug by something called as lycopodium spore method. I remember initially I've just told you about this. Lycopodium spore means you will mix your sample along with spores, and when you observe under microscope, each spore will have a unique shape which you can count. And your sample, for example, ginger, if you're mounting, ginger has starch. If you add iodine, you'll be seeing purple color grains. So you can count the number of starch grains, you can count the number of lycopodium spores, and there is a particular formula. If you apply that formula, you'll get how pure your ginger sample is. So percentage purity can also be found out by microscopy. Now coming to leaf constants, can you name the leaf constants?
Leaf
Palace
isolate number.
number, Alfred ratio.
Stomatal number, stomatal index, palisade ratio, vein islet number, and veinlet termination number. Okay, so these five are constant. For example, Senna has a stomatal index of 13.33%, means if you analyze, it will be having around that particular value only. Then you can tell your drug is standard. Okay, so otherwise, the general microscopies, you're going to take a transfer section or powder, you'll be mounting and observing under microscope. What are you going to observe? You're going to observe various cells and tissues. So what are the types of cells you can observe? What are the types of three basic type of tissues which you are going to observe?
Parenchyma, collenchyma,
Good.
and sclerenchyma.
Parenchyma, collenchyma, sclerenchyma. These three will come under ground tissue. Dermal tissue also you will observe. See, there are three types of tissues what you will observe. Dermal tissue, ground tissue, vascular tissue. Okay, dermal tissue, ground tissue, vascular tissue. Vascular tissue, what do you observe?
Xylem and phloem.
Xylem and phloem. Ground tissue, you're going to observe parenchyma, collenchyma, and sclerenchyma. Which is living, which is dead in among these cells?
Sclerenchyma is dead.
Sclerenchyma is dead. So when you add lignin, it will take a pink color stain. Parenchyma and collenchyma are living. Parenchyma is mainly meant for support. Okay. And there are different types of modifications. Parenchyma with air is called as aerenchyma. Parenchyma with chlorophyll is called as chlorenchyma. So like this, different things you have. Sclerenchyma is dead and it is mainly for mechanical support. Okay. Now coming to first one, dermal tissue. In dermal tissue, what do you observe? Dermal means what? Yes.
Outer surface.
Surface. So in the surface, what do you expect to see? There will be epidermal layer. In case of younger plants, if it becomes tree, epiderm would get converted to periderm, where you can observe cork, phellogen, phelloderm, etc. Otherwise, you see epiderm, and in between epiderm, you can also see stomata and trichomes. Clear?
Yes, ma'am.
Yes, ma'am.
What is stomata now? What is stomata?
Opening on the surface of the plant
What
for
is its use?
as an inlet of gases and
Okay.
water vapor.
So Stomata is a minute epidermal pore present on the surface of plants, specifically leaves, stems, etc. And this is mainly meant for exchange of gases. Okay, transpiration and exchange of gases. How many types of stomata are there?
4
Four
types.
types, ma'am.
Not 4, it is 5 types of stomata. What are the types of stomata you know?
Parasitic, diacytic, and anisocytic.
Good, Pallavi. 2 more?
Anesthetic.
Actinocytic.
Nomocytic.
Anemocytic. Very good. So, depending upon the arrangement of stomata with the epidermal cells, we have 5 types of stomata. So, that is uh, um, parasitic, diacytic, anisocytic, anemocytic, and actinocytic. Parasitic, as the name tells, the stomatal pore is parallel to the epidermal cells. Diacytic, stomatal pore is perpendicular to the epidermal cells. Aniso means what? Aniso means? Iso means what?
Same.
Same. Aniso means unequal. Okay, so one stomata may be smaller, one may be bigger. Aniso means unequal celled stomata. Anemocytic means irregular cell, irregularly arranged stomata. And actinocytic is nothing but radial. Like a flower, you'll be able to see central, you'll have epidermal, central, you'll have stomatal pore. Surrounding it, you have like a flower epidermal cells. So there are 5 types of stomata: parasitic, diacytic, anemocytic, anisocytic, and actinocytic stomata. Now, what are trichomes? And how many types of trichomes are there?
They are the tubular elongated epidermal cells.
Okay. How many types of trichomes are seen?
2 types, ma'am.
3 types.
Not 3, 2 types. What are the 2 types?
Glandular and
Good, correct. You are
Covering.
very confident.
Covering.
Covering trichome and glandular trichome. So covering trichome, it will be somewhat like inverted V. Okay, you have a base and on top you have a pointed apex. Glandular trichome is nothing but like a gland. So this can store volatile oil, resins, etc. Main use of covering trichome is nothing but protection. These are nothing but hairy like projections present on the plant surface. If you see lady's finger, no bendi, you can see hairs on it. Those hairs only are covering trichomes. So glandular trichomes, it will be like gland. Now again, depending upon the type of cell, depending upon the number of stalk, the number of head you have, unicellular, seriate trichome, bicellular, uniseriate, multicellular, like this you have so many types of trichomes. Fine. So in microscopy, you're going to observe all this. Okay, you're going to observe different types of tissues, dermal, ground, and vascular tissue. Next, in xylem and phloem, you'll be observing what is the type of arrangement: collateral, conjoint, open collateral, or closed collateral. So all these types of arrangements you're going to observe in microscopy. Fine. So coming to the pharmacognostic scheme, you're going to start with synonym, biological source, family, morphology, microscopy. Next is after microscopy, what do you study?
Chemical constituent.
Very good, chemical constituents. So whenever you're writing chemical constituents, you should write the class first. For example, alkaloids. In alkaloids, put a dash and write quinoline alkaloids. Then cinchona. Okay. Name will be at the end. You should first write the main name of the secondary or primary metabolite, then the class it belongs to, and finally the names. Then you should draw structure of single important constituent, and finally you will give other important other constituents present also. Clear? Now for example, coriander contains what? What is the metabolite coriander contains?
Lanolin.
I want you to tell me the primary or secondary name of the primary or secondary metabolite. Anything with good smell has what? So something which has good smell has which type of metabolite? Does it have alkaloid?
Flavonoid.
Flavonoid is anything with color, orange, brown, anything with color is flavonoid. Anything with good smell is, think of plants which have good smell. Name the plants which have good smell.
Lily.
Rose.
Pallavi is with lily. Think of the spices what is used at home only.
Cinnamon.
Clove.
Clove,
Cinnamon.
then cinnamon,
Cardamom.
coriander, fennel, all cardamom, all these have which constituent? Name the
Tannin.
secondary metabolites.
Alkaloids.
Alkaloid,
Glycosides.
glycosides.
Tannins.
Yes, then
Flavonoids.
tannins, flavonoids, then which
Volatile
one is
oil.
made up of
Volatile oil.
Yes, volatile oil. So something which has good smell will have
Volatile oil.
volatile oil. Something which is bitter to taste will have
Alkaloids.
alkaloids. Good. You should remember like this. Anything with good smell will be having volatile oil. Anything which is bitter in taste will be having alkaloids. Anything which is astringent in taste will have. Anyone knows the answer for this? Anything which is astringent in taste? Astringent means like pomegranate. As soon as you eat pomegranate, you feel a sort of taste, no, sort of numbness. That is called astringency. So anything with astringent taste has. Nobody knows the answer? Okay. Anything which is astringent will be having tannins. So if tannin is present, its use will be astringent. Okay. Anything which has astringent taste will have what?
Tannins.
Tannins. Okay, tannins. Fine. So now coming back to the chemical constituents, you're first going to write, for example, coriander. Mahantesh was telling the example of coriander. So coriander first you should write volatile oil. In volatile oil, it has D-linalool, Linalool. D-linalool is also called as coriandrol. Okay, then draw the structure of coriandrol. Then you should write it also has other volatile constituents and certain carbohydrates, resins, etc. Like this, you should write the chemical constituents. Okay, fine. Coming back to the scheme again. Synonym, biological source, family, sometimes geographical source if it is required. If your syllabus says you have to write about geographical source. Then comes morphology with diagram, microscopy, chemical constituents, then chemical tests if any. Okay, next one is if there is any specific chemical test, it has to be mentioned here. Next, after that, yes. What is the last one?
Uses.
Adulteration.
Uses. Good. Aishwarya, good. Shashank also good. First it is uses, followed by substitutes and adulterants. Okay. So uses, how do you write? You should write it as 1, 2, 3, 4. Now, although we know coriander is having volatile oil, definitely it will be aromatic, flavoring agent and stimulant. You should not write this as first use. First use will be its pharmacological use. So first you will write it as carminative, second aromatic, third flavoring and flavoring agent, flavoring and perfuming agent you can write. So first will be its main use. Okay, first you're going to write carminative. Its pharmacological use should be written, followed by aromatic, then flavoring agent. Clear? Last is substitutes and adulterants. What do you mean by substitutes and adulterants? Quickly, we'll end the class. The last one we are discussing now.
Adding similar looking waste products are
Okay.
without any
Good,
contents.
Shashank. Yeah, good. It is nothing but debasement of an article, or you're adding something else which is going to spoil your product. That is adulterant. Substitute means it can be of some other species. Okay, like for example, cinnamon. If we take, there are two types of cinnamon. You have cassia cinnamon, and also you have cinnamon calisaya. Two types are there. So one is just like normal flat pieces, and one more, it would have formed quills. Okay, so this two is got from different places. One is got from Sri Lanka, one is got from some other place. But if you look at the constituent, some may have more amount of cinnamaldehyde, some may have less. But then in case of substitute, we are not going to spoil the main product. By mistake, it would have got replaced, it would have got substituted. Clear? Adulterant means you're totally adding something else which looks similar to the drug, but it will not be having the chemical constituent at all. See, whatever we talk now, mainly it is on basis of chemical constituent. In an adulterant, chemical constituent only will not be there. For example, if you take cardamom, rodent rodent fecal active constituent at all, isn't it? Did you get the difference between substitutes and adulterants?
Yes,
Yes, ma'am.
ma'am.
Okay, substitute means it may have the constituent, the amount of chemical constituent may be less or maybe more, whereas in adulterant, active constituent only will not be there. It is going to reduce the quality of your drug. Fine. So this is the pharmacognostic scheme of the drug. From Monday's class onwards, we will start with the introduction, and then I'll allot each one of you a topic. If possible, to one of you, I'll mail the PPT, the empty frame of PPT based on the pharmacognostic scheme. So you can start adding, or maybe one drug I'll be able to teach you. You can see and you can make your slides as interesting as possible. So you will be evaluated for this and marks will be given for this. Clear?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Now quickly, let us finish off. Tell me the pharmacognostic scheme fast. So you're going to begin with?
Synonym.
Synonym.
Yes. Next.
Biological
Biological
source.
source.
Next.
Condition, part of plant,
Family.
family.
Next.
Morphology.
Okay. Before that, geographical source also you can add. Geographical source, morphology, then
Microscopic.
Microscopic evaluation.
microscopy. Next.
Chemical
Chemical
constituents.
Chemical evaluation.
Yes. Next.
Specific
Chemical
chemical tests.
Tests, if any. Then
Uses.
Uses.
yes. Next.
adulteration and substitution.
And adulteration.
Substitutes and adulterants. Very good. So please write this scheme in your book today. You can write it in your tutorial book as well without seeing. Okay. I want all of you to by heart this particular scheme. This will be very in this particular unit. Next unit you have 11 drugs followed by 10 drugs. So totally you have around 51 drugs in your syllabus. Okay. So for all this, the scheme is same. It is very easy. Yes. Tell me.
Ma'am, physical evaluation will not come in this, no?
No.
Like
Physical
moisture.
evaluation not required. Only when you're doing the morphological analysis as per IP. Okay, if you're doing the monograph analysis, then it is required. Otherwise, we generally don't need it. Okay,
Okay, ma'am.
what standards they would have given solubility and all that, it is not very important.
Okay.
Clear?
Yes,
Yes, ma'am.
Yes, ma'am.
In between, no, I'll just ask one of your classmates. Anyone can. I think Pallavi, you can call me. I'll just tell you how to make a table. Okay, like that table you can neatly make and share it in the group. All of you make a table like this, and then you can just write the names of drugs this week and leave it. Next week onwards, slowly you can start filling the drugs and you can start by hearting. For example, I'll tell you the first one will be serial number, second is common name, third is biological source, Fourth is family. Fifth is chemical constituent. Sixth column is uses. So if you make like this, it'll be like a ready reckoner. Whenever you have time, just go through it. 50% of your syllabus will be complete. Okay. Uh, uh, Anybody got an idea how I told you? Can you make a rough column and show it to me? Just
Yes,
you
ma'am.
can turn your tutorial book landscape, okay? Landscape, and just write on the heading as crude drugs. First column will be serial number. Next is common name. Next, biological source. Biological source, you should leave more space. One of you just make one column like this and post it in the group. If any changes is required, I'll tell you. If not, all others in your tutorial book, you can make like this. So slowly you can start writing next week. Clear?
Yes, ma'am.
Okay, so I'll conclude the class now. Have a nice day. In between this weekend, please practice the biosynthetic pathways. Okay?
Okay, ma'am.
Bye-bye.
Thank you, ma'am.
Thank you.